Good afternoon, class. In today's session, we are going to, or I'm going to teach you, or you're going to learn about a very new topic, which is very relevant to today's time. And the title of that, or chapter is called as Monoclonal Antibodies, DNA Diagnostic System, and Molecular Diagnosis of Genetic Disease. First of all, let's understand what is a diagnosis. So diagnosis is a process by which we determine the cause of the disease. The cause of the disease. Now, this diagnosis is very important because if diagnosis is not solid or good or accurate, then the treatment will not be effective, right? So a clinic or a hospital which has a good diagnostic system will also have a good treatment outcome. And if there is a mistake in the diagnosis and the treatment is not right, then there is a problem for that system. Now, things have changed considerably in terms of diagnosis. So what used to happen before is that generally many hospitals or clinic, they used to have a associated lab. This lab, let's say, would work, or a pathology department, let's say, for a hospital or a big clinic system or a nursing home. They used to have a good laboratory support or a pathology department and to get the report in-house. But now there are now big companies like, for example, Quest Diagnostics, right? Who can take the blood, who can come to the clinic, take the blood or any specimen and give the results quickly and in 1 day, 2 days, depends upon the complication. And there are also companies that can take frozen tissues and they can give the diagnosis with regard to biopsy or something. So the idea I'm telling you is that diagnosis is changing. And now what is happening that, these companies, they have a lot of data. Companies like Quest Diagnostics, they have a lot of data. And that data, so understand this they have the data from us, from this population, right? So they have a lot of statistical information already in their data bank, right? And they can use that data to develop artificial intelligence, right? For example, let's say there's a patient who comes from, it's now, if you realize in today's date, it's very difficult to distinguish between allergy, asthma, and bronchitis because of very similar overlapping symptoms. There can be chronic allergy, which will look like asthma, and allergy can cause wheezing. Asthma also causes wheezing, bronchitis, pneumonia. So they're very related. Now, let's say people, they get some results based upon respiratory test or blood test, then they can quickly use their in-house data and the patient data to match what this particular patient could have. What is the high risk for this? So diagnosis, through this example, what I'm trying to communicate to you is that through this diagnosis, the field of diagnosis has undergone a big revolution. And that's because of data storage, and that is growing, right? For example, let's say in past 1 year, these companies, these hospitals, they have tons of data on COVID-19. And at some point of time, when they analyze their data, they can predict, many things. Let's say there are still there are various SARS virus or pandemic. So diagnosis is going to change. And I think I shared one article in the beginning about looking at imaging or MRI and uh, to diagnose breast cancer or any other cancer, and that will facilitate the because artificial intelligence, you're matching your data with millions of patients, so the chances of missing a diagnosis is lesser and lesser, and starting a good treatment is more. So there will be improvement in treatment. But the surprising thing is that the diseases are also changing courses. The heterogeneity in the disease is increasing. For example, in microbial infection, antibiotic resistance is increasing. Uh, in terms of virus, there are viral variants, and these viral variants, they evolve under immune pressure. So there will be always an ongoing challenge. So again, the diagnosis has to move hand in hand with the therapeutics. So today's class is dedicated to molecular diagnosis. Why we are using molecular diagnosis? Because if you notice, the diagnosis is mainly, let's say, in terms of anemia. In anemia, what happens? A patient can come who is feeling weak, and the doctor says, okay, the physician says, let's go for a blood test. And the blood test, you get hemoglobin level or RBC count below threshold. Let's say you get a RBC or hemoglobin count, number of 6 to 7, which is anemic. Now, this anemia could be because of bone marrow suppression. This anemia could be because of low iron intake. Maybe the person is not eating well. So, so to make the diagnosis very comprehensive, we have to go at molecular level, right? To understand the disease pathogenesis, because all these biomolecules, proteins, carbohydrate, lipids, right, DNA, they are they're the same, right? What you study as in biochemistry, proteins, carbohydrate, lipids, and they also become the marker for diagnosis. For example, in anemia, one would like to know the counts of hemoglobin. You would want to look at the shape of RBC, right? So cells, and cells would be in sickle cell anemia. Let's say the shape of the cells, RBCs change. So what determines the shape of a cell? Of course, the mutation, but cell shape is also determined by um, lipid, because lipids are the major component of the membrane. So then diabetes, insulin. What is insulin? It's a protein, right? It's a hormone, so it's a protein with an activity. Uh, Metabolic diseases, right? So the diseases, the diagnosis has to be done at molecular level to know the correct picture, what is going on inside the body of a patient. So, but this class is focused more on antibodies, right? So, so let's go back to the first slide. So in this class, this chapter focuses on molecular diagnosis using an antibody. Now, what is an antibody, right? Before I go into antibody, I just want to briefly describe to you what is an antigen, right? So if you see, but before I come to these slides, I would just go and talk a little bit about the antigen. So what is an antigen? Antigen is anything foreign in our body. Anything foreign. It could be a pollen, it could be a dust particle. So when these foreign entities, foreign agent comes to our body reacts, right? human body reacts, right? And this reaction could be protective in nature, or it can accelerate a disease process. For example, that's what you see in allergies, right? We get the pollen, and then a pollen can cause sickness of various kinds, from sneezing to wheezing to fever, right? So anything that is foreign, that can activate. When I say human reaction, what I actually mean here, to be precise, I mean human immune response, human immunity, right? And what is human immunity? Human immunity is essentially mediated by a bunch of cells and proteins. I'll come back to this later. Human immunity, right? Now, a little bit of complication. So, just pay attention what I'm saying. Not all foreign entities that makes an entry into our body can cause immune reaction, right? So those foreign, so anything that is foreign is an antigen, but anything that is foreign entity may not generate immune reaction. Those foreign agents that can generate immune reaction within our body, those are called as immunogen. Right? That's the difference between an antigen and immunogen. An immunogen uh, will always cause an immune reaction. But an antigen may not always cause human immune reaction. So anything that is foreign is antigen, but anything that is foreign may not be immunogen, because body may not react. Now these antigens could be foreign, it could be a pollen. Sometimes what happens, our 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 body can also initiate reaction. So let's say um, so a foreign antigen could be a foreign, an antigen could be from inside the body. So that's what happens in autoimmunity, where our body reacts to our own cells or own proteins. So coming back to again to the slide. So this is a brief introduction I wanted to talk to you about the antigens. So antigen, when it enters into our body, it elicits the immune reaction. I just want to make sure that we are recording. So I'll just pause and then just start the recording again. Okay. So as I mentioned to you, the antigens can give rise to human reaction. And one of the reaction is immune response. And one of the branch of immune response is antibodies. And these antibodies are proteins or glycoproteins. Now this slide shows you the shape, the structure of an antibody. So if you see this structure is just like a Y-shaped structure. If you focus, this is a Y-shaped structure. So if I write the letter alphabet, if I write the alphabet Y, so for example here, right? So if you look at this alphabet, this shape of antibody is just like Y. But that's for sake of understanding, for simplicity. But if you look more detailed, you will see that there are two chains of proteins which are longer, and there are two chains of proteins which are smaller, right? Two chains longer and two chains smaller. So this red arrow shows a longer chain, right? This is called as heavy chain. And there is a shorter chain. This is called as light chain. So based upon the size of these proteins, a typical antibody is composed of a heavy chain and a light chain. Right? So that's the structural knowledge of an antibody. Now, this, if you look at the alphabet Y, right? If you look at the alphabet Y, you have this this tail or stalk or stem, right? This is here heavy chain. This is called as constant region of the heavy chain, constant region. And then you have this, this V-shaped here. If you look at the V-shape, this V-shape creates a pocket, right? A pocket here. So you have a pocket here also, right? Just like Y. Now, this pocket, V-shaped pocket, this is the place where an antibody binds to a foreign substance, which is an antigen, right? This is the antigen binding region. But how does it bind? This binds because of these two, heavy and light chain. So this together, this structure is called as antigen binding site. Antigen binding site, right? Now let's go into more details, right? So let's analyze this figure, right? So this sort of represents that three-dimensional structure of an antibody. So now antibody formation. So in our body, there are millions of antibodies, right? Some antibodies are in the blood. Some antibodies are housed in the lymph node, right? And it depends upon the presence of foreign agent in our body. So if you zoom into this figure, so this, I won't go into the detail, but this is the organization of an antibody. So this is the heavy chain. This is the heavy chain. This heavy chain is made up of a constant region, the stem region, and the variable region. And then there are light chains. That light chains also compose of a variable region and the constant region. VL stands for variable region from the light chain. VH stands for variable region from the heavy chain. And then there are constant domains within this antibody. So long story short, antibodies are proteins which have a very complex secondary structure. First of all, made up of heavy and light chains. And then these individual heavy or light chains, they have a variable region and the constant region. This is the region which is the most important, because this regions, they varies from antigen to antigen. If this don't change, our body will not be able to react to millions of. So let's say in this time, our human body is dealing with what? COVID, the season is changing, pollens, and flu season, so many things. So this variable region is most important because this gives a lot of plasticity to the antibody molecule to fight to the foreign substance. Now, which cells produces antibodies, right? In human body, which cell produces antibody, right? Antibody is a product that is produced by special cells. That those cells are called as B cells. Right? I won't go into the details. So, but I just want to show that look at how our body is planned. So remember in the last class, I talked about stem cells, right? Self-renewal, self-replication, self-differentiation. So these B cells which produce antibodies, they emerge or they develop from human hematopoietic stem cells. Then they become pro B cells, then large pre B cells, and small pre B cells. Then they release early antibodies. These are called as IgM. I won't go into the detail. And then these B cells mature, right? In inside mother's body, liver. This is a very common question in MCAT, where antibodies or B cells are antibodies are produced in an infant who is inside the mother's body. So it comes from fetal liver. So first, fetal liver, produces B cells, and B cells produces antibody, right? But as the fetus, the infant become independent, the role switches from fetal liver to hematopoietic stem cells. So that's very interesting, right? Part of human physiology. Now, a little bit more information about B cells. So some B cells could be naive B cells. Naive means they haven't seen any foreign object. They haven't met any foreign objects, and the presence of these naive B cells will be maximum in the developing humans inside the mother's body. Now, the most important B cells, which are important from the vaccine point of view, they're called as memory B cells. These B cells are already exposed to the antigen. But what our body does, just like we type something on the computer or your term paper, we save it, right? And then you can, if your computer is running fine, you can come back and take out that data, which could be 5 years, 10 years back. Now you have iCloud. You can take out, as long as the your saving profile is good. So there's a memory in a computer also, right? So our B cells also has memory B cells. These memory B cells are stored in lymph nodes, right? Such that when our body encounters the same pathogen again, these memory cells will spring into action, right? So naive B cells, which are fresh, they haven't met any antigen, and the memory B cells are just like army of cells waiting for an attack. And the moment the antigen comes, they are ready to. So, and then there's a path, right? So there is naive B cells. The naive B cells, when they see an antigen, they become activated and they produce antibodies. So there are various kinds of antibodies that I won't go into the detail. In an activated B cells, these B cells get activated by the pathogen or foreign or pollen, and they can secrete. Secrete means release. Antibodies are released from the B cells. They can be of IgG type, IgE type, IgA type, and IgM type. And then the final stage of B cells is called as plasma cells. These are the mature B cells that secrete antibodies, right? And I want to emphasize that we'll come to know later, that these plasma cells, there's a disease called as multiple myeloma. In multiple myeloma is a disease of plasma cells, where a patient's body keeps making a specific type of antibody. So the cancer of plasma cells is called as multiple myeloma. And in these, a certain plasma cells get selected and they keep making antibodies. So if there is an excess amount of antibody in our body, it starts reacting to human tissues and causes cancer. But what scientists have done, they are using this plasma cells. So these myeloid leukemic cells, multiple myeloma cells, they're immortal cells to produce antibodies again and again. So these immortal myeloma cells, they are being used by the scientists to make monoclonal antibodies that we'll talk later. Right? So let's go to the next slide. So with these basic information, let's go more. So as I mentioned to you, antigen, an antigen is a foreign entity that enters into our body and tries to fight our immune system or to cause disease. Right? So an antigen is a foreign substance. And this antigen can be a big antigen. For example, let's say pollen. And this is surface of a pollen, for example. So this a particular pollen can give rise to 7 kinds of antibodies. So this is, let's say, the structure of a pollen. An antibody first could be directed towards this part of the antigen or pollen grain or to COVID-19 virus. Antibody 2 can be geared towards this domain of this antigen, could be pollen or COVID-19, and antibody 3. So that the point is, any foreign antigen that enters into our body can generate several kinds of antibodies. Right? Now, why I'm saying this to you? So what I'm trying to say that although this patient, let's say, can generate antibodies to this particular, let's say, COVID-19, but they are still not able to fight the infection and people are getting sicker and losing life also. What does it mean? That although we have antibodies, but antibodies that can neutralize, neutralize means make the virus ineffective inside our body to cure our body is lacking. So although an antigen, a foreign substance, can elicit 7 kinds of antibodies, but it is possible that out of 7, none of them are helpful. That is happening in HIV. And that's why we don't have a good HIV vaccine until now, because we don't throw neutralizing antibodies. We throw a lot of antibodies, but we're not throwing the therapeutic antibodies, right? In COVID-19, that yes, we know that spike protein is notorious. So if there's an antibody against the spike protein, that patients can be treated. That's why plasma therapy is being strongly considered. So coming back, that an antigen is a complex substance that can generate a range of antibodies. Right? So now I would introduce to you one more term, that antigenic determinants. So let's say these are the 7 determinants on this antigen that can generate 7 different type of antibodies. So don't get confused that all the 7 antibodies are similar. No, they are not similar. They're different. Why? Because they are reacting to the different surfaces of the same antigen. Right? So antigen is a foreign substance. A foreign substance that generates immunity is called as immunogen. Not every antigen can generate immunity. And an antigen has a complex organization. It can throw several kinds of antibodies, and that are called as antigenic determinant. So what happens that let's say this is a B cells, and this B cells has this antibody. This antibody can recognize this pathogen. It could be virus, bacteria, or pollen, and then try to generate immune reaction to fight this pathogen out of our human body to make us disease free. So antibodies, they can, on one hand, they can bind to the pathogen. On the other hand, they can bind to a cell to create a networking. So networking will help to to take a stronger step to neutralize the pathogen. So in an antibody, the variable region, the pocket I told you, the V-shaped pocket, it binds to the pathogens. It binds to the pathogen. Now, now think about it in cancers, right? So cancer cells, they have different kinds of antigens on their surface. For example, in ovarian cancer, these ovarian cells which have grown to become cancer, they can throw up a special antigen called as CA125. So CA125 is not present in a normal situation. But in ovarian cancer, this CA125 could be a marker. So an antibody can be made against CA125 in ovarian cancer, and this antibody can generate battery of immune response to cure a patient with cancer. And very fast, the field of immunotherapy is growing very fast, because each one of us have our own capacity to fight any disease. It could be a viral infection, it could be allergy, it could be cancer. So knowing the personal repertoire of immune system is very important. So now here, that there are two kinds of antibodies. Right? This is very important. One is called as monoclonal antibody. The name itself suggests mono. Mono means one. Clonal means a battery of millions of cells that emerge from one cell. So monoclonal means the antibody is being produced by only one cell, and this will recognize only one antigenic determinant. So let's say, to make it more simpler, let's consider that this is an antigen. Right? This, what I'm showing here to you, is an antigen. This is an antigen. And what you see in this picture, there's only one place this antibody is binding. So this is an three-dimensional structure of an antigen, which is mostly alpha helix, and this antibody is binding to one particular spot. So these kind of antibodies are called as monoclonal antibodies. Now, how it is possible? It is possible because all the cells are emerging from one clone, and all these millions of cells that are emerging from one cell, they are binding to one spot on the antigen. Now, in contrast, let's see this. If you see this structure now, in contrast, okay? If you see this structure, you see here that this is an antigen, but antibodies are binding to various places on the same antigen. Here it is one, but there are several antibodies binding on the various positions in the same antigen. These are called as polyclonal. Poly means more than one or more than two, right? So each antibody, it emerges from one clone, right? So this is one clone of antibodies. These are several clones, right? One clone, this could be an MCAT question. One clone can give rise to how many types of antibodies? So one clone can give rise to one type of antibody. Here, what you see here is that how many types of antibodies are here, right? Against one antigen, you see 1, 2, 3, 4, 5, 6. So there are 6 clones of cells that have reacted to produce antibody to this antigen. So this becomes polyclonal. antibodies are emerging or being secreted from different clones of cells. But in here, there's only one antibody, type of antibody. This is emerging from, that's why it is monoclonal, emerging from one clone of cells. Clone that is derived from one cell. But there are advantages and disadvantages of both. Monoclonal antibody could be best for diagnosis, right? Or a treatment also. But these days, polyclonal is becoming more important. Why? Because of variants, right? A monoclonal antibody in a variant can miss. Let's say this portion of the gene mutates and the shape of the protein is changed. This antibody will not bind. I repeat this concept. Let's say this antibody is binding to this section of the protein. This section of the protein is being made by a section of DNA. So if the DNA for this section of protein, this domain of protein, mutates, right? Which is happening in case of many virions, viruses. Then, if it is mutated, then the shape of the protein will change. Then this antibody will not bind. So that's a disadvantage of monoclonal antibody in terms of variants. But if you see here, if there's a polyclonal antibody, it is possible that, let's say, this is being missed because of mutation, but all these will target all these. So there are 6 different clones of antibodies. There's a mutation in one spot. Okay, there's a mutation, so this antibody will not bind, but at least 5 others will bind, and they can combat the virus, right? So with regard to the emergence of variants, polyclonal antibody could be more reliable for treatment. So this slide shows the difference between the polyclonal antibodies and monoclonal antibodies. So polyclonal antibodies is relatively cheaper to produce because you don't care about how many clones you get. It could be 2, it could be 6, it could be 20. But monoclonal antibody is very specific. You have to have only one. So it's very expensive to produce monoclonal antibody as compared to polyclonal antibodies, right? And for the production of monoclonal antibodies, remember, I talked about a disease called as multiple myeloma. So the plasma cells in multiple myeloma are immortal cells. Those plasma cells from the multiple myeloma that is being used to produce monoclonal antibodies because they are immortal cells. They recognize only one epitope. So let me go back to this concept here. The part of antigen to which a human or an organism throws antibody is called as an epitope. In monoclonal antibody, there is only one epitope. In polyclonal antibody, there can be multiple epitopes. So that's what this slide is showing. That polyclonal antibody recognizes multiple epitopes. It could be from 1 to 100. But a monoclonal antibody, the beauty of monoclonal antibody is that it recognizes only one epitope. So that's how the name comes, monoclonal. Okay. So as I mentioned to you, a monoclonal antibody is generated by, so this is a slide that briefly says how a monoclonal antibody is generated. So we take the spleen cells, which produces a specific types of antibody, and we fuse with the myeloma cells. So when you fuse spleen cell that produces specific kinds of antibody with the myeloma cell, these cells become immortal, and they can be selected to get a particular kind of monoclonal antibodies. Right? So once again, spleen cells are the cells which produces B cells, which has B cells that produces antibodies. They can be fused with the myeloma cells, which are immortal plasma cells. Remember, I told you plasma cells in what context? I told that plasma cells are those cells of mature B cells that secrete antibody. So when you fuse these two cells, they will keep making antibodies which are of monoclonal nature. Here, the selection becomes very tedious. Somebody who is working in the lab generating monoclonal antibodies has to really do a good job with the selection of the right cells. Now, let me give you an example. So in abnormal breast cancer cells, in this, so breast cancer is a complex disease, right? It's heterogeneous. It depends upon the stage, but there are some breast cancer cells which are positive for this protein, HER2. And they, on their surface, they express this HER2 proteins. So if we make monoclonal antibodies against HER2 in this breast cancer, then this monoclonal antibody, if injected into these patients, they can generate immune response, and this antibody can work as a missile to kill the breast cancer cells which are HER2 positive. And this is giving very positive results with regard to the increasing the lifespan of breast cancer cells. So antibodies can be used for diagnosis, antibody can be used for therapeutics, antibody can be used for research. So I just wanted to a little bit talk about another kind of cells called as T cells, right? Or the different kinds of cells which can be present in our body. They're the natural killer cells. They try to kill the virus or bacteria right away when they enter. But sometimes they don't do. Most of the time they don't do because these viruses, these bacteria, they're more smarter than our immune system. Then there are memory cells, which tries to remember. Memory cells is the backbone of vaccine development, right? So we are in the process of taking vaccine. The whole country is getting vaccinated. What is the goal? To create these memory cells such that it prevents the future infection from COVID-19, right? And you can also argue with me that, Professor, if you talk about memory cells, why we are taking flu vaccines every season? So the answer to this is that flu virus, they keep mutating under the immune pressure. So each season, so the new variants will be not recognized by the old memory cells, right? So our memory cells for a flu virus, influenza virus, is as good as last season. In the next season, it is mutated. In next to next season, again mutated. So these memory cells may, but most likely may not, counter the new strains of flu virus. That's another virus strategy to trick human system. Okay. Now, antibodies can also be used to conjugate to drug. For example, let's say there's a monoclonal antibody that can target prostate cancer cells. So first, we know the antigen that distinguishes the normal prostate cancer with the disease. I repeat, we need to know the protein that distinguishes normal prostate cells with the prostate cancer cells. Once you know that protein that distinguishes the normal prostate cells with the prostate cancer cells, we can develop antibody to those prostate cancer cells. We can make this further effective if we conjugate this antibody for, let's say, radioisotope or with cytotoxic drugs that can, let's say, activate P53, which is a tumor suppressor protein. So from one bullet, you are attaining two goals. From one stone, you are hitting two things. So you are hitting the cancer cell with twice. One with antibody that would generate robust immune reaction against this cancer cell. Also, you're targeting the cytotoxic drugs, for example, doxorubicin, or let's say you're trying to develop. So there are second generation prostate cancer drugs which are anti-androgens. So you can have this antibody against prostate cancer cells to that protein, and also tag with second generation anti-androgens. So you have two in one strategy to destroy a cancer cells. So and in many countries, thyroid is a problem, because of not good quality of life, or not good protein and not good health level, and lack of iodine that causes thyroid problem. So you can generate antigens from this to develop antibodies to develop a in-house kit to diagnose the problem with thyroid. Now, I would like to switch gear regarding the diagnostic part of the antibodies, right? So diagnostic means antibody. So this is an antibody. This antibody should be able to detect the antigen. This antigen could be virus, bacteria, or any other fungus, parasites. So let's say this is a patient's antibody. So let's say this is my blood, right? My own blood, and I'm trying to see whether I have antibody to COVID-19. So I'll be drawn, somebody will take my blood out, and will take the fluid from the blood. And then if I have antibodies to this COVID-19, so antibodies are very, very specific molecule. It will right away bind to the COVID-19 if I have an antibody to COVID-19. Okay, that's first step. So this is immune protein antigen antibody complex. Now what will detect antigen antibody complex? This antigen antibody complex will be detected by a secondary antibody. And this secondary antibody will be coupled to HRP. Now let me go back and just give you some little bit of explanation. So let's say this is an antigen, which is a virus, right? So what will detect this antigen? It will be detected by an antibody. So this is an antibody. But the question comes out, what will detect this complex? So what scientists have done, that they have developed special kind of antibodies, right? And this antibody, they have coupled with an enzyme. This enzyme is called as horseradish peroxidase. Horseradish peroxidase. In short form, this is called as HRP. Right? So you develop, so on one hand, this is one that you have antigen antibody complex. Right? And to detect, you need another system which is called as secondary antibody, second. Now this antibody, what it will do, it will recognize the constant chain. Remember I told you that there are two parts to an antibody. First, this part, the V thing, which recognizes the antigen, and the constant region of an antibody. So this secondary antibody will bind to the constant region. Constant region. So now what is happening? So there is a complex here. What is a complex here? Number one, you have an antigen, and then there is a patient's antibody, right? Then there is also, you have a secondary antibody. Right? And this secondary antibody is labeled with an enzyme called as HRP. Right? So this secondary antibody, this will bind to this complex. Why it will bind? Because this can only bind to FC region, constant region of an antibody, FC region. Now, how do you detect this circuit? So this circuit will be detected by this enzyme. So in those patients who have antibodies, this circuit will be completed, right? And then this enzyme will work. So all we have to do is to add a substrate for this enzyme. And if this complex is made, this circuit is there, then this enzyme will give color. This technique is called as ELISA, enzyme linked immunosorbent assay. So you have the patient's antibody. If this patient is positive, this will bind to an antigen, and this will be this antigen antibody complex between patients and antigen will be detected by a secondary antibody, which is coupled with an enzyme called as horseradish peroxidase. And all these are done in a 96 well plates. And now scientists can really miniaturize this assay, and instead of 96 well plate, they can do it in a let's say 1,000 well plates. So at one go, they can screen thousands of patients. And this is the technique called as ELISA. What I spoke to you is the basic concept for ELISA. Now, another technique that is used for where antibody is used pricelessly is called as Western blot. In Western blot, what happens that in contrast to doing ELISA, so ELISA is done in a 96 well plate that I'll show you later. ELISA, so a 96 well plate, if you haven't seen, is something like rectangular plate, and there are 96 wells in this plate. 96 wells, sorry. There are 96 wells in this plate, going from this end to this end. There are 96 well plates, so one can screen 96 patients. 96, each well will correspond to a new patient. In Western blot, what is done is that the antigens are separated. So let's say there is I won't go into the detail. The antigens are separated on the basis of molecular weight. And this is a membrane. And then what is the advantage is that the patient, through this test, you can recognize against which protein. So a virus, a COVID-19 can be made of 10 or 20 proteins. Through Western blot, you can know that patient is throwing antibodies to which specific protein. So when Western blot is a more specific and sensitive test as compared. ELISA is more sensitive. ELISA can predict more false positive and can miss also depending upon the antibody titer. So, but Western blot is very, very specific. Right? So in Western blot, it is done with the help of on a membrane, whereas ELISA is done on the well. ELISA is more sensitive, whereas Western blot is very specific because you're looking at against a particular protein, antibody against a particular protein. Right? So I might have antibodies to A, B, C, but I may not have antibodies to the neutralizing antibodies for COVID-19. So here we can look at the profile of antibodies that a patient might have. So let's say if a patient has less of neutralizing antibody, there are high chances that that patient can get reinfected. In contrast, if a patient has a high level of neutralizing antibodies to COVID, which suggests that the patient can fight COVID-19 next time also. Why? Because he or she will have the right memory cells in the lymph nodes so that the same virus infects, it can be fought over. So that's the advantage of Western blot. Now, there are some diseases which are very, very complex, such as malaria, right? Malaria is a parasitic disease. It is very common in tropical countries. And the complication is arising these days because of drug-resistant mutant. And the malaria parasite is moving from blood to brain. So it is very difficult to conclude whether the headache in a patient is because of meningitis, of it because of sudden, let's say migraine or malaria. So the disease spectrum. So what is the classical feature of malaria? The classical feature of malaria is that to have evening chills and fever. But now because of drug resistant or mutation or polymorphism, the pathogenicity of these parasites are changing, right? So how can we detect? We need more robust diagnostic kits to detect these kind of diseases where the organism is changing with time under the pressure of immunity or because of antibiotics or because of such as malaria. So the DNA, so right now we talked about antibody-based, therapeutics, diagnostics using ELISA and Western blot. Now there are DNA-based diagnostic system where we cannot distinguish whether it is malaria or whether it is typhoid. So you need the help of molecular biology because they have different genomes. So we have talked about in earlier classes, PCR could be good for those organisms which have genetic material as DNA, but for organisms like COVID or HIV, which has RNA as genetic material, you need to do RT-PCR, right? So in malarial parasites, where there are no good antibody response, and so malarial parasite is a biphasic organism, right? It spends some of its life in the mosquitoes, salivary gland, and also in human. And in humans, it can be in the liver, it could be in the RBC, it depends. And in each different cells that the malarial parasite homes, whether it is mosquitoes, salivary gland, or gut, or human liver, or human red blood cell, the shape changes, right? And so how to detect these things? On top of it, there are mutations that are changing the disease style. So the DNA-based diagnosis, such as PCR, becomes very, very important. Sometimes we need combination of these factors. So, I've been trying to learn myself and to share with you, about COVID-19, and I'm taking, and you might have noticed that I'm taking out every opportunity to talk about this, right? COVID disease. So spike protein is very important for COVID-19 infection, right? And this spike protein can bind to the ACE receptor on the human cells. And that's how it makes entry. Now, if you make an antibody, and this is a neutralizing antibody, so see the circuit here, spike protein binding to human ACE receptor, lock and key concept, they like each other. That's the starting point of the infection. But if somebody has, antibodies, good antibodies, whether it comes from vaccine or his or her own, then this antibody can interrupt the interaction of spike protein with the ACE receptor. That's the whole mission. That either we need a memory cell that would develop this quickly antibody that would interrupt here, or to have a monoclonal. Remember, few people got, they are getting antibodies also as a treatment, polyclonal monoclonal antibody. This company in the upstate New York, Regeneron, they are making, I think they're making a poly or monoclonal, I have to verify. So these antibody can interrupt the spike protein binding to the ACE2 receptor, right? And if all these antibodies, they bind to the virus all over the surface, the virus can no way be active in our body, right? So they can't spread. So that's the goal of the vaccine, that's the goal of the monoclonal antibody, that's the goal of the plasma therapy, that is the goal of the polyclonal antibody. But look here, in this figure, antibodies are only developed after second week in case of a new infection, right? If there is no, so depending upon the immunity, right? If this patient doesn't have enough neutralizing antibody, so despite he or she has antibody, cannot fight the infection, right? So has to be monitored, right? So this gap in the infection, and the emergence of antibodies, so we are giving almost 1 week to 10 days, at least 1 week, depends, 3 days, 4 days, 1 week, 10 days, free hand to our virus. So the moment COVID-19 infects us, we are giving the virus free hand for at least 1 week. Go ahead, replicate in our body, because antibody is coming in the second week, after 10th day, right? So the DNA base, the RNA base, the molecular biology becomes very important. That's why the RT-PCR kit becomes important. And that's how the vaccines become important. Because the moment immune cells sees, so vaccine is very important. And today I was reading somewhere, I'm still in touch with my mentors, my teachers from my PhD days. So one of my PhD teacher, mentor, he shared that some people are very hesitant about the vaccine. I'm not supporting vaccine. This is a class of molecular biotechnology. I'm not anyway discussing pro or against vaccines. But some people are not very comfortable with vaccine because they think that some people are having blood clot formation in their body, in the body, so better not take a vaccine, right? But my teacher shared that 1 in 600,000 will have a symptom of blood clotting in their body with vaccine. 1 in 600,000. But at least 1 in 125,000 will, of course, die of lightning. So the chances of dying out of lightning is more than taking the vaccine. He's a virologist, my mentor, and he thinks that So again, so this phase, which we are giving free hand to the virus, only thing that can protect us is vaccine, because we don't know. The carriers are asymptomatic, right? They could be healthy, they could be sick. We don't know. Most of them are asymptomatic, right? So this is the portion that we need to cover ourselves. And the only thing that can cover ourselves technically is vaccine. So again, how ELISA works for COVID-19 system, just to review. So there can be an antibody on this plate. We put the target antigen, which is the patient's blood, which carries viral antigen, spike protein, right? So if the patient's blood has spike protein, it will bind to the antibody. And how will you develop this antigen antibody complex? Because you can't see this in the 96 cell plate. So you need an additional tool, which is called as secondary antibodies. This is primary antibody that captures the antigen, and this is the secondary antibody. So to develop, to know whether this circuit exists, because this circuit will tell you that a patient is positive or not. If there is a color because of this enzyme activity, the patient is positive. If it is negative, means patient is, if the color is negative, means patient doesn't have infection. If the color is positive, so which means, so with this, we end this chapter on molecular diagnostics, and I am open to questions.